At this point, we are on topic 6 of the Biology Keystone Review, and the topic covered is about DNA and its processes, and this should help you answer questions 24, 28, 29, and 31 in your Keystone Review packet. So what we need to do really quickly is refresh on the idea of nucleic acids. We did talk about nucleic acids in our first podcast when we talked about the macromolecules, carbs, lipids, proteins, and here we are, nucleic acids. So you can see here's a picture of the nucleic acid structure. We had those monomers, those building blocks of each one were called nucleotides. Each nucleotide had 3 parts to it, and you can see they're shown here in green, red, and blue. We have a phosphate group, which is shown here with a circle, and here is our P and OH group. We have our 5-carbon sugar, and there's actually going to be some slight differences in that 5-carbon sugar, which we'll discuss. And we have a nitrogenous base. It's called a nitrogenous base because it has less nitrogen atoms in it. There are a couple different variations or types of nitrogenous bases that make up these nucleotides that in turn would make up nucleic acid. In addition to just talking about nucleic acid, we're really talking about 2 main types of nucleic acid, and you can see them up here. We have deoxyribonucleic acid and ribonucleic acid. Deoxyribonucleic acid is also known as DNA, and a little bit of information about it. It's a double-stranded molecule. So basically what happens is those little nucleotides, those monomers, get strung together into one chain, and then what happens is there are basically 2 chains that wrap around each other. DNA is often referred to as that double helix, if you've ever heard that term before. The reason it's called DNA is that 5-carbon sugar is called deoxyribose. Basically, it's missing an oxygen compared to its other friend. We have 4 nitrogenous bases. You have an adenine, a thymine, a guanine, and a cytosine. And basically, they're just slightly different in structure. Those slight differences in structure are important, though, because they lead to the fact that certain types of bases will bond with others. And the way that I personally remember this is the ones that begin with straight letters, T's and A's, they always go together. They're buddies. So you can see every time there's a thymine on this half of the double strand, there's always going to be an adenine on the other half of the double strand. So you can see T with A, T with A, T with A, T with A. Now, the T's don't always have to be on the right. They just happen to be like that in this picture. And then your other nitrogenous bases, your curvy letter ones, your G and your C, your guanine and cytosine, they always go together. So you can see here cytosine goes with guanine, cytosine with guanine, cytosine with guanine, and so on and so forth. We're showing you here D is your deoxyribose and P is your phosphate group. So what scientists will often do is they'll call this back portion the sugar phosphate backbone. Another really important thing to notice here is in between these nitrogenous bases, in between these A's, these T's, these G's, and these C's, you see teeny little dots here, which are supposed to show you hydrogen bonds. Now, we talked about hydrogen bonds in that very same first podcast where we talked about some of the different types of bonding and properties of water. Hydrogen bonds, again, are weak bonds. They're not really true chemical bonds. They're more weak associations between polar molecules. That is so important that they are actually in between here as we talk about some of these later processes. Oh, besides deoxyribonucleic acid, we have ribonucleic acid, which is also known as RNA. Couple of differences. Where DNA is considered a double-stranded molecule, has 2 strands, RNA is a single-stranded molecule. Instead of having deoxyribose as its sugar, it has ribose as its sugar. And you'll notice the nitrogenous bases are mostly the same. We still have an adenine, and a guanine, and a cytosine. However, we have uracil instead of thymine. So over here, if my A and T are always going to go together, in this case, my A and U are always going to go together. And here's just a picture of RNA. Again, you still have your phosphate group over here. You still have ribose, just not deoxyribose. It's still your sugar. And you can see you have your different nitrogenous bases. Your adenine, your guanine, your uracil, and your cytosine. They would pair up to another half of a piece of DNA. With our ribonucleic acid, or with our RNA, we have 3 different types, and each type has a slightly different function. So we have one that looks like this, and we call it messenger RNA. And we call it messenger RNA simply because it has the message of DNA to be carried to the ribosome. And we'll explain that more as we talk about transcription and translation, but just that's kind of where the M comes from. We also have this guy, who is transfer RNA, or tRNA. And tRNA basically transfers things, hence its name. And what it does is it takes this amino acid, which you can see right here, and it transfers it to something called a ribosome. And we talked about that in the Khan Academy video, with ribosomes being the protein synthesizers or protein factories. And what amino acids are used for, we talked about that in the first podcast. We're putting some of this information together. Amino acids are the building blocks or the monomers to proteins. So transfer RNA is going to take these building blocks for proteins. They're going to transfer them to a ribosome, who's going to kind of link them up together through dehydration synthesis to actually make a protein. And then our last type of RNA is ribosomal RNA. It's part of a ribosome. Ribosomes are really made out of RNA and protein. And the function of ribosomal RNA is it's going to be actually reading the message on mRNA, and it's going to be making or assembling a polypeptide chain from amino acids. So there are three things we can do with nucleic acid, whether it's DNA or RNA. We have a replication, which we're going to talk about shortly. We have transcription, and we have translation. So if you look at the central dogma, it's just kind of a way to say we have this nucleic information, and there's different things we do with it in our body, and pretty much every single organism on this planet uses this genetic code. So replication, basically you're taking a strand of DNA and you're making another copy. Transcription, you're taking the message in DNA and you're putting it into an RNA format instead. And then lastly, translation, you're taking this message that's on our piece of mRNA and you are translating it into the language of proteins via using a ribosome to kind of string those little amino acids together to make a protein. So what we're going to be doing is we're going to be talking about each one of these in a little more detail. Replication is, again, the idea you take a strand of DNA and you basically kind of Xerox it. You want that copy to be completely identical. So what we have to do is DNA is, again, a double-stranded molecule. We're going to snap those two strands apart or kind of unzip them using an enzyme. And then what we'll do is we'll have another enzyme come in. Remember, enzymes are proteins in our body that carry out chemical reactions. And the enzyme is going to notice it has a naked half. And so what the enzyme is going to do is it's add new base pairs in. And then basically anything gets tidied up, cleaned up if there's any errors, and we get two identical strands of DNA. Oh, this is a very basic diagram of what's going on. There are all sorts of players in this game. There's a DNA helicase, a DNA polymerase 1, 2, 3. There are topoisomerases, there are SNRPs, and all of this other stuff. We're going to keep it nice and simple. So what we have over here is, you can see this is the strand that hasn't been unzipped yet. They call them parent strands. They are a double-stranded DNA. The DNA strand gets unzipped, and you have basically a half here that needs another partner. An enzyme would come in and say, hey, I see a thymine. Because of base pairing rules, I know my straight letters go together. Thymine goes to the adenine, and it's going to bring in an adenine. And what we'll do is we'll start joining these nucleotides together to make, at the end, two new strands. Each strand is identical to the original parent strand from which it came. That's just basically replication. You want to make the exact same copy. Our next idea is transcription. With transcription, what you want to do is you want to make a mRNA version of DNA so that we can use it to make a protein. So what we're doing is we're making a strand of mRNA based on a specific segment in DNA. You don't want to copy the entire chromosome. When you're making a protein, you're really looking for a little piece right here that codes for something. It could code for your hair color. It could code for your eye color. It could code for your height. It could code for your susceptibility to cancer or Parkinson's. Your DNA pretty much codes everything about you. And what we do is we again have to unzip that DNA. We need to break all those little weak hydrogen bonds. And then we have an enzyme come in. And what that enzyme is going to do is it's going to notice if there are any strands here on just the one side, the template strand. With transcription, we don't care about this other side. We kind of just ignore it. And what we do is we just make a piece of mRNA that would match. And then what happens is your DNA rezips like nothing ever happened. And then if it's a eukaryotic cell, like you and I, then that mRNA is going to leave the nucleus. If it's a prokaryotic cell, it's all done in the cytoplasm. It just kind of scoots out of the way. Oh, here's a picture. Again, this is a very generalized summary. There are lots of enzymes and lots of things going on here. We're just kind of looking at the overview of transcription. So my blue is my strand of DNA. It's a double-stranded helix. And what happens is wherever the message is that I want to kind of make a new version of, it gets opened up. And that's what this teal thing that kind of looks like a guitar pick is. It's an enzyme. And so it splits it open. And what happens is I'm adding my base letters as they match. And then once I make the piece that goes with it, the DNA just kind of re-zips behind it. So T goes with the A that's down here. So T and A. But remember, we don't have a T in RNA. So instead of putting a T here, A goes with U. T goes with A, C goes with G, G goes with C, A goes with U. And it will continue until it gets to a spot that it kind of knows, hey, we're not going to code here anymore. We do need to talk about this genetic code. So we keep saying things like A's, T's, and G's and C's, or adenine and cytosine. Or in the case of RNA, we have A's, U's, G's, and C's. Those letters, they actually mean something to a cell, and they mean something to a ribosome, and they mean something to different types of RNA. And what we have here is something called a genetic code. And the genetic code is looking at 3-letter segments, and we call them codons, of pieces of mRNA. And these 3 little segments, or these codons, they actually code, or they match up with a specific amino acid. Remember, amino acids are the building blocks of proteins, and the whole goal here is to actually make a protein. So if I look at this wheel, there's a couple of really important ones to look at. Right here, A, U, G. Okay, so the way you read this wheel, it's the first option, your second letter option, and then your third letter option. So for methionine, A, U, G. Methionine is really important because we call that our start codon. Whenever a ribosome sees A, U, G, it knows it needs to start making a protein. There are 3 others that I want to look at, and that will be UA, U-A-A, UAG, U-A-G, and UGA, U-G-A. Those are stop codons. Stop is not a type of amino acid. Stop simply means stop making a protein. So what happens is just like AUG says, hey, it's time to start making a protein, start to find these amino acids and put them in place. UAG, UGA, and UAG, UGA, and WAG say stop, we're done making a protein. But again, any of these other ones, GGG, that's glycine. It's just one of the 20 amino acids. CCU, protein. AGA, arginine. They are just building blocks of proteins. Here's just another diagram because, to be perfectly honest, I'm not sure what type you'll see on the Keystone exam. In case there are one of these questions, I want to make you comfortable with it. In this case, 1, 2, 3, 4, those are your first choices. 1, 2, 3, 4, that would be your second letter choice, and 1, 2, 3, 4, this would be your third letter choice. So again, you can see UAA, stop codon, UAG, stop codon, UGA, stop codon, and AUG, methionine, your start codon. So translation is a big one. We're going to be using all 3 types of mRNA. We're going to be using mRNA, rRNA, and tRNA, so all 3 types, to ultimately create a protein or a polypeptide chain. So what happens in kind of a summary is you have your piece of mRNA, and it goes into the ribosome, and it gets fed through the ribosome, and the ribosome looks for those 3 letter segments, those codons. And what it does is it finds one, and then it looks for the tRNA. The tRNA that would go with it. So just like there are all these different codes for each one of these amino acids, there are all these different types of tRNA. There is a tRNA for proline, which is one of the amino acids. There's a tRNA for methionine, which is one of the amino acids. There's a tRNA for histidine. There are tRNAs for each one of those little codons to find the matching amino acid. The only tRNA molecules that don't have a pair are the stop ones, because you wouldn't want anything there. It's just supposed to stop the message. Ultimately, what happens is we keep joining together those monomers, those amino acids, and then we get a polyprotein or polypeptide, a protein done at the end. So in terms of pictures, we're looking at something like this. I have a piece of messenger RNA, and that was transcribed in the nucleus if it's a eukaryotic cell. This piece, you can see right here, AUG, there's my codon for start, goes into the ribosome. So what happens is the ribosome goes through and it sees that it needs a methionine. These transfer RNA, these tRNA molecules are floating all in the cytoplasm, and each one has its specific amino acid attached to it. So what happens is it's like puzzle pieces. Well, hey, the start codon looks like this. This is the complementary or matching sequence that goes with it, and it's got the right amino acid on it. And what happens is it drops off its amino acid, it reads the next 3 letters. So UUC, that codes for this amino acid. AAA codes for this amino acid. And we kind of keep shuffling on down from left to right until it hits, and you can see right here, UGA, one of those stop codons. When it hits the stop codon, the whole business is done, and this polypeptide kind of changes shape a little bit, folds, modifies, and turns into a protein. So just kind of a basic overview of these processes. DNA can just be replicated and turned into DNA. DNA's message can be transcribed into a piece of mRNA, and the information in a piece of mRNA can be read by a ribosome, and with the help of tRNA molecules turn into a chain of amino acids or a protein. However, this process is not perfect. When most people hear about mutants, they think about X-Men, or they think about horrible things that can happen to our body, such as polydactylism up there. The person has 6 fingers on each hand instead of 5. Not all mutations are bad, and that's something really important to think about as we start our future podcast talking about evolution. Mutations can be very beneficial to a species for survival, but we always tend to think of them as bad. So what you want to do is you want to think of mutation as this. It is anything or any change to the sequence of your DNA. Your DNA is supposed to have some order of A's, T's, G's, and C's in it. Anything that causes that to change, that would be a mutation. Mutations can be caused by things outside of your body, such as radiation. We know there's a huge link between skin cancer and UV radiation, and there are also chemicals. There are things like all the chemicals in smoking. We call them carcinogens because they are also linked to cancer. Cancer is pretty much just too many mutations acquiring in your body. However, besides just big bad cancerous mutations from chemicals and radiation, in our bodies, whenever we go through the process of replication, which happens in the S phase of interphase for the beginning of cells to divide and mitosis, whenever we go through transcription and translation in order to prepare to make proteins for our body, there are places where errors can happen. We have error checking messages and systems in our body to make sure that A's only go with T's or A's only go with U's and G's go with C's. But sometimes those enzymes that carry out the chemical reaction, they goof up. And it happens. Sometimes the mutations aren't that bad. Sometimes the mutations can be really, really bad. So what we're going to look at right now is just called a point mutation. Point mutation means it happens to only one letter, one nucleotide. So you can see right here, they're showing you your piece of DNA. And then what they're showing you here is this is how it would normally turn into mRNA if it were translated, because T goes with A goes with U. And then if it were transcribed, this is the amino acid chain that would result. You can see here in this substitution, though, what happened is that somewhere along the way, maybe when it got replicated, this cell screwed up, and instead of having a C, they have a T. So when that piece of DNA gets transcribed into mRNA, that 3-letter segment, that codon is now different, and it codes for a different amino acid. So this is methionine, histidine, threonine, and leucine. It's a different chain. Sometimes it might not make that much change in the shape of a protein, so the chemical reaction isn't changed. Remember, proteins are all about their shape. You screw up the shape of a protein, it won't be able to function. Sometimes they can actually be more beneficial for an organism. Or this mutation could be a huge bad thing for the organism. Even more scary than a substitution mutation would be one of these two guys. And this is the idea of a deletion or an insertion. What happens, again, we have our normal code here, is that somehow a whole extra base pair gets thrown in the mix. And the reason that this is so bad is that everything downstream, everything after that mutation gets shifted off by 1. So remember, we read in sets of 3 letters. So what happens is everything after that gets completely messed up. So you can see the mRNA code is completely different than what it should be up top. And the amino acids, methionine now is isoleucine, arginine is now alanine, thyramine is now tyrosine, and leucine happens to be the same there. But this starts getting really messed up. What can also happen as a result of this is the stop codon is now gone. So this protein just becomes this ginormous thing. Or the stop codon happens right away and it's this teeny little snippet of a protein that can't do anything. To make it a little clearer, you can see here, instead of using our RNA letters, or A's, U's, G's, and C's, this is a simple sentence with 3-letter words. The fat cat ate the rat. If I have a deletion here, and the H is gone, now the sentence becomes teft, ata, tet, her, at. It means nothing. So the whole frame got completely ruined as a result. The whole protein is different. And then lastly, we have chromosomal mutations. And when we talked about our cell division one, we talked about chromosomes being huge pieces of DNA that get separated in cells as they divide. So what you're looking at here is this is a chromosome broken into segments. So a deletion, you can see the B one just absolutely gone. There could be the gene for causing an eye to form. So if that organism happens, they wouldn't be able to form their eyes. You could have a duplication over here. In a plant, it could be an extra thing. So maybe it's now they produce twice as much fruit. It could be good. Or it could be something where you produce too many toxins and the organism kills itself. An inversion here, instead of being A, B, C, D, E, F, it flip-flops pieces. And this can be really bad because if there's certain coding or a certain gene, a certain piece of information that was right between A and B, that can get all sorts of messed up. And then lastly, we have a translocation. So this would be 2 different chromosomes, non-homologous chromosomes. And what happens is they break and swap. And this causes all sorts of bad news. A lot of these things are often fatal for organisms. That being said, they can also lead to interesting changes in evolutionary paths and evolutionary history. They can be beneficial a lot of the times, however, they aren't. Oh, to sum everything up, we have our DNA, and our DNA has our genetic code in it through the letters, those nucleotides, and it allows us to have instructions or a blueprint to make all the important proteins and functions and structures in our body. What we do is we have our cellular division. We need DNA replication so that our cells can contain that code from cell to cell, from person to person, from generation to generation on down. DNA can be transcribed into mRNA, and then that mRNA can be translated into an amino acid, into a protein sequence for our body. And then lastly, this process can go wrong, and it can be due to all sorts of things, but these processes of replication, transcription, and translation can lead to changes in our genetic code.